how important the genetic variability for improvement of vegetables, and for that centers of origin, germplasm collection and conservation is also uh, important. So let us study as part of that introductory chapter, centers of origin, germplasm collection and conservation. So cultivated plants are not distributed uniformly around the world. That means anywhere you go, same kind of variability you won't get. Some part of the globe you will get maximum variability. Some part you don't get any variability for a given crop. So for that, where that variability is existing, a concept of center of origin was proposed by Pavilo. So who was heading plant. Oh, Leningrad plant industry, it was headed by Pavilo. Um, uh, the concept of centers of origins. That means some crops have originated in a particular place. He also postulated the law of homologous series in variation. Characters found in one species also occur in other related species. So thus the diploid, tetraploid, and hexaploid wheat show a series of identical characteristics. So that is, his finding, that law of homologous series in variation. That means any species, it throws the variation, but if it evolves into next, so that when it evolves, there are some characters which are common. So the characters found in one species also occur in other related species. That's the the law of homologous series in variation. It explains. It is not that suddenly you change the species, but there will be some common characteristics. So that is the variation is spread across the related species. So N.I. Webilo, he proposed that the crop plants evolved from wild species in the areas showing diversity and termed them as primary center of origin. Fortunately, it's a primary center of origin because normally the crop species means what we cultivate, those plants, right? Some plants we may not cultivate, we cannot call them as crop species. Crop species, they have originated at some place where their wild species are there. From that they have evolved, they have originated. And where you can find them, it is the place where you find the diversity, variation. So that place, so the wild species in the area showing diversity from where the crop is originated, and that place is termed as the primary center of origin. So in some areas, certain crop species show considerable diversity of forms, although they did not originate there. There is also confusion that some places you will find the variability, but still they are not originated there. Such areas are referred as secondary center of origin. So, you should remember these people like Webilo. You have decandle. Lot of, the germplasm collection has been done from him earlier. Decandle collected germplasm in crop species. And the many times the references you will get under his, N.I. Webilo, the brilliant Russian botanist and phytogeographer, was a global thinker. Because of his difficult childhood growing up with famine, he focused his efforts and great energy towards improving the human conditions. So many of these great people, they faced difficult situations and they worked towards that. From Charles Darwin's idea, he realized that plant species were not fixed. They evolve over time. They are not static. They will change. So that's what we term it as evolve. They evolve. The Darwin's book, Variation of Animal and Plant Under Domestication, it actually significantly influenced this Wabilov's thinking and his studies. So this book, what Darwin wrote on variation of animals and plant under domestication, it introduced the concept, the little particles of inheritance that Darwin termed the tangents. He used the term tangents. Something is getting inherited, or what Hugo de Vries shortened to genes, right? Pan genes to genes, you call. That was also the thinking when the book is written. There are things which get small little particles, they get inert from parents to progenies. Three of his significant theories, the Ni Wabilos, the law of homologous variation is one. His contribution, what the variation found in one species is also found in related species from where it has originated, are closely related. Then the center of origin for cultivated croplands. Cultivated, what the crops which are useful to human being, we call them as a croplands. So where they are originated. And the concept of genetic erosion also. This is not just where they are originated, but some amount of variation is lost, genetic erosion. So this greatly influenced the subsequent global plant science breeding and conservation efforts. Why this is important, you should realize, the most of the plant breeding is utilization of the existing variability. Where the variability is there, this concept of the uh, centers of origin, or subsequently turned as centers of diversity. So initially, Vavilov proposed 5 centers of origin in 1924, which developed into 8 in 1935. Although his final papers, 1992 to 97 posthumously published, discussed 7 major centers with minor additions for there. So Vavilov's centers, 8. Vavilov centers accounted for most of the major crops of global economic importance. Uh, in that period of time, what the plants he has considered, they are important. He missed a few significant crops in North America, such as sunflower, and new crops such as blueberry, Vaccinium corybosum, and strawberry have globally developed since his time. So after that, these other crops have added there. Otherwise, whatever the 8 centers of origin he proposed, that is sufficient. So Wavilo, what he proposed 8 centers of origin, let us just look at the Chinese center, that is the East Asian center, center of origin for, actually Chinese center of origin refer, or East Asian center, one and the same. It's a mountainous region of central and western China and the neighboring lowlands, Korea, Japan, and Taiwan. This is the native region of soybeans, millets, many vegetable crops and fruits. So we have estimated that 20% of the world's cultivated flora originated from this Eastern Asia. And the primary center, crops which are having primary center, soybean, radish, and several species of allium, rako, brassica, brinjal, some species of cucumis and cucurbits, like muskmelon. Then secondary center, somewhere it is mentioned in the literature, French bean, cowpea, turnip, and then others. Sometimes while referring, they don't refer it as primary or secondary center, but just says that this particular species belongs to that particular center of origin. Other crops like coleus, and the second center is the Hindustan center of origin. This includes tropical India, Indochina, southern China, and islands of Southeastern Asia, like the Burma, Assam, Malaya Archipelago, Java, Borneo, Sumatra, and Philippines. Some names are changed of now, but this excludes the northwest India, that is Punjab. This is native uh, is native of land of rice, right? Sugarcane, tropical fruit, and vegetables. So this Hindustan center of origin. It's a native of the rice, sugarcane, tropical fruit and vegetables. More than 30% of the world's cultivated plants are originated here. So 20% if you give for the Chinese center, 30% is Hindustan center origin. It's almost 50% it is covered here. Um, Primary center crops like coffee, brinjal, cucumber, lactica indica, that is Indian lettuce, Diascorea, certain species, Indian radish, Raphanus indicus, and other category, if you put it, snake gourd, winged bean, elephant foot yam, smooth gourd, dolichos bean, drumstick, Indian spinach, muskmelon, pointed gourd, amaranth, ash gourd, basella, bitter gourd, ridge gourd, roselle, snap melon, checker munis, cluster bean, coleus, colocasia, curry leaf, right? Roselle, that hibiscus, and then the snap melon here we have like Cucumis sativus variety, Mammonia. Then checker munis, right? Multivitamin. And then the third center he proposed is Central Asia, that is Inter-Asiatic Center or Afghanistan Center. It includes the interior mountains of Asia Minor and then um, um, Iran, Syria, Palestine, Transjordania, Inner Asia, and Northwestern India, that is Punjab, Kashmir, and all comes here, Central Asia. So Pakistan, all of Afghanistan, Tajikistan, Uzbekistan, and Tianxia. So native crops include wheat, rye, and fruit trees. About 15% of the important cultivated plants for the world are originated from here, right? So primary center crops, like peas, broad bean, radish, muskmelon, carrot, onion, garlic, and spinach. Others include bottle gourd, mustard, turnip, Welsh onion, and that. The fourth center is Asia Minor Center of Origin, that is Caucasian Center. It is also known as the Near Eastern or the Persian Center of Origin. It includes Asia Minor, Transcaucasia, Iran, and highlights of Turkmenistan. It includes original species of temperate fruit trees, additional species of wheat and rye also found here. Primary center having crops like carrot, cabbage, species of allium, lettuce, and secondary center mentioned in literature, turnip, black mustard, and others like beans, beets, sprouting broccoli, chive, kale, currat, onion, pea, Brussels sprout. So then the fifth center is the uh, Mediterranean center of origin. This includes the countries bordering on the Mediterranean Sea. Great ancient civilization, selected many vegetables and forest crops, including the olive and carob tree. Around 10% of the species of the cultivated crops originated here. Pea, broad bean, brassica species, cauliflower, cabbage, turnip, onion, garlic, beetroot, lettuce, artichoke, asparagus, celery, kale, nolcol, Swiss chard. Then the sixth is Abyssinian center of origin. It includes Ethiopia and the hill country of Eritrea, South Africa, right? And it produced a seed oil plant like and the coffee also, right? So these are some of the crops. Probably about 4% of the world crops originated here. Primary center crops like pea, onion, and okra. Secondary center broad bean. Then other group crops like wattle gourd, celery, cucumber, coffee, fenugreek, watermelon, West Indian gherkin. So the seventh center is Central American, that is Mexican center of origin. Central American center of origin. It includes southern part of the North America, Mexico, and the West Indian islands. Staple plants such as maize, cotton, beans, pumpkin, cocoa, avocado, and subtropical fruits originated here. About 8% of the important crops of the world originated here. So the primary center having crops like corns, French wheat, lima bean, muskmelon, pumpkin, sweet potato, chilies. So others like amaranthus, chow, jack bean, squash, and tomato. So the eighth center, what he proposed, the South American center of origin, that is Andean center, you can call Andean region, just like the mountainous, Himalaya is there in India. Andean mountainous region is there in South America. It includes the Andean mountain range covering regions of the countries like Peru, Bolivia, Ecuador, Colombia, and parts of Chile and Brazil, and whole of Paraguay. This land produced many tuber-bearing crops such as potatoes, pinene tree, and the coca bush. So primary center, many species of potato, corn, lima bean, cucurbita maxima, tomato, and cassava originated here. So if you just look at here, the primary, the centers of, you can see here, the Central America, and this is what the southern um, South American center of origin. And of course, right from the beginning, we have got, some mention like Hindustan center, right? Uh, Asian center. This is Abyssinian center. This is Mediterranean center. Of course, again, further division and some additions have been made. So what the Webulo 1992 been adopted from Webulo. So later in 1935, Webulo recognized these centers of origin, and he split the Hindustan center into two, and South America into three, and added one new center, USA center, making totally 12 centers. Only made it. So that Hindustan center was divided into Indo-Burman center and Sia Malaya Java center. So Indo-Burma, you find the crops like snake gourd, winged bean, elephant foot ham, smooth gourd, dolichos bean, drumstick, Indian spinach, muskmelon, pointed gourd, amaranthus, ash gourd, Ceylon spinach, that's bacilla, bitter gourd, ridge gourd, roselle, snap melon, chackermunis, cluster bean, coleus, colocasia, curry leaf, cowpea, and brinjal, cucumber, Indian lettuce, Indian radish, and the escoria. And the Sia Malaya Java is an ash gourd, breadfruit, colocasia, curry leaf, elephant foot ham, and ants you can find. So then South America, he divided, South America Central into one, Peru, Ecuador, Bolivia, where chili, corn, French bean, lima bean, potato, pumpkin, squash, tomato, Zatusoma. And then for second one is the Chile Center, that is potato. And the third one is the Brazil, Paraguay Center, tapioca. And 12th center he added, that is USA Center of Origin, Jerusalem artichoke, that is Helianthus tuberculosis, and globe artichoke belongs to this particular center of origin. So finally, 12 centers, I think conveniently, almost all the food crops, majority of the food crops are covered by these 12 centers of origin. So the centers of origin, instead of calling them as the centers of origin, we, many times the confusion, you are finding some crops at two different centers of origin. Then where it is exactly, the originated, you are not sure. It's very difficult to say. Although you say where the variability is more, where it has originated. Where the, not only that criteria, even wild species where they are, the related wild species, then you start believing that this is the place where it originated. So conveniently, it is better to refer them as the centers of diversity than the centers of origin. Whatever, right? These are the the centers of origin of crop species, definitely 12 centers. You don't find various other parts of the continent you will find, but you don't find the crops originated there. So, yeah, for crop improvement, it is where the variability is there to know. It is very important. Then you have to collect, conserve, and utilize. So then only your crop improvement. I tell you, more than 70% of the efforts go for the utilizing the existing variability. Only 25% you can say that we create variability and utilize. This very important. That means collection of germplasm is so important. Unless it is totally exhausted, it is not wise to go for creating variability. Simply use it first and then. So germplasm collection. What do you mean by the germplasm? It is the sum or total of hereditary material or genes present in a species is known as the germplasm of that species. It is the sum or total of hereditary material or genes present in species is known as the germplasm of that. So how do you get that? By collecting more number of plants. Germplasm collection is collection of large number of genotypes of crop species and its wild genotypes. Then only the total hereditary material present in that species you are having in your collection. More, genotypes you have to collect. So that is the germplasm collection. So germplasm means it's a total sum or some hereditary material present in that species. Then germplasm collection centers are known as gene banks. If you are having some places where you are maintaining, after collecting, you are maintaining, that place is known as gene bank, which includes the genotypes from all over the world, right? So it is important that regular crossing with wild species is required, because if you disturb, if you collect from their existing place where they are originated are now showing the maximum variability, and you try to maintain them. Gene bank is located some other place. You don't have the continuous accessibility for the wild species, right? It's a large area. Gene bank is a very restricted place, small area, and more collections you are having, and you may not give the the full opportunity for the species, other wild species also to grow with that. So it is important that regular crossing with wild relatives is important component of evolution, right? This is not never, it is not never ending, process. It's continuous process, even future also. Germplasm collection furnish the richest source of variability. It's a small area you are having maximum collection. That's what I said. Crop improvement would ultimately depend upon the availability of the variable. Otherwise, yeah, these are again the centers of where this the locations of administrative office for the gene bank, centers for the consultative group of CJR, right? Agriculture research, where you can find Summit, Mexico, then Seattle, Colombia, then Lima in Peru, and Biodiversity International Rome, Italy, ICARDA, Beirut, Lebanon, and Africa Rice Center, Cotonou, Benin, IITA, Ibadan, Nigeria, the World Agroforestry Center, Nairobi, Kenya, and ILRI, Nairobi, Kenya. You have the place, and the ICRISAT, Patancheru in India, and IWMI, Batarmulla in Sri Lanka, World, the fish, Penang, Malaysia, CIFOR over Indonesia, then IRRI, Los Banos, Philippines. So these are where headquarters for CJR. It's a group which it funds the various the crop improvement activities around the globe. With the modernization of agriculture, it has led to large tracts of lands are put under the pure line and hybrids, and this has led to gradual disappearance of local varieties and land. So that means we are losing, because of this human intervention, more, if forest area, the natural habitat is allowing, the species keeps on evolving with the intercrossing with the wild species and all that. But you are totally clearing the ground, and you are bringing the land area under the crops where a single variety or a single hybrid or a pure line is grown in a large area. So that has led to disappearance of some local varieties and also land races. Cultivation and grazing in these, it's not only that, it's the natural habitat also you are disturbing, allowing the animals to graze there. So cultivation and grazing in their natural habitat has led to gradual destruction of wild species and their breeding grounds. If you lose that, the evolution is going to be stopped. You don't have answer if you don't allow this evolution to happen. You don't have the answer for the big problems you are going to face in future. So wild relatives may get eliminated by introduction of weedy species. That's one more thing. Why this is all happening? The weedy species means they are highly adaptive and they compete with the useful species for humans, right? Useful means it's useful to human, and those species can disappear. So the gradual loss of variation in the cultivated and their wild relatives is referred as genetic erosion. The loss of variation. It's not immediately it's going to happen. It's a gradual, slowly. There is a loss of variation in the cultivated and their wild relatives. It is termed as genetic erosion. It's a concern today. Genetic erosion, the as per the International Union for the Conservation of Nature and Natural Resources, IUCN, about 10% of the world flowering plants, that accounts to 20 to 30,000, are under the threat of extinction. These many numbers are, they may disappear. Extinct means totally, disappear. You don't find anything, no recovery process. Extinct means complete disappearance. So that is the threat because of all these human interventions. The process responsible for genetic erosion, why this is happening? The breeding methods, like, most of the time we are following the inbreeding. One type good, only that you allow it to multiply in a large area. And of course, selection you make it. What is that? Something not useful, you will reject it. You'll throw it, you will cull it. But that whatever you have culled or thrown out can be, the answer for the future problems. So that is really threatening. And then advanced agriculture system. It's not only our breeding methods itself, of course. Then the advanced agriculture systems like replacement of land races, completely, the new advanced varieties, land races disappeared. Germplasm is lost, and invading the marginal areas or natural habitat. Some areas where the breeding grounds are there for some species, you are bringing it under cultivation, or you are allowing your animals to graze there. That is destroyed. They are losing. And then clonal multiplication. Clone. What do you mean by the clone? Same thing is multiplied. Then other things are displaced. So that is also responsible. So this is the threat. And then what you have to do? Integrated gene management. It's very important. In situ, community conservation, and ex situ. So in situ means, say, in your lab, the national parks, protected areas, biosphere reservoirs, and then World Heritage Sites. Like forest, environment, and wildlife departments, they are taking the responsibility of that. Community conservation, some people are involved in situ on farm, land races, folk varieties. Farmers themselves can do it, and tribals and rural families, they can do it. And there also ex situ on farm, that is sacred groves. If you have, you preserve some seeds in your home, okay, that becomes situ. Ex situ means some, no, not in the form of seeds, but the plants are there, trees are there, you are maintaining it for a longer period. That is the kind of ex situ. And then, of course, this is community conservation. And then exclusive and ex situ, that is botanical gardens, zoological gardens, gene banks. You are allowing those species to be there, cultivated there. And annuals, you have to, replant every often. Whereas perennials, you have to maintain those trees continuously. Gene banks, government agencies and universities can do this. This is the effort, it is required. Well, so now you call that, yes, the gene pool, right? This is the collection of the accessions. There you can classify these whatever the accessions you have collected into primary gene pool, secondary gene pool, and tertiary gene pool. So primary gene pool means it includes all the strains of the concerned crop species. Crossing between the members of GP is easy. Primary gene pool, first, they are easily crossable. Whereas secondary gene pool, it includes all the species that hybridize with. Whatever you have collected, you are saying hybridize with the members of primary gene pool. What the group is there, first one, they can get hybridized with that, with some to considerable difficulty. It's not easy. Yes, you can do it, but there are difficulties, incompatibility between the members of GP1 And the hybrids are at least partially fertile. Sometimes you may have fertility problem also if you cross with GP1. So there may be a ploidy difference, chromosome alterations, or genetic barriers. So gene from transfer from GP2 to GP1 are possible, but usually difficult, right? That is secondary gene pool. And the tertiary gene pool you group, the species belonging to this group represents the extreme outer limit of the potential germplasm, and the species that are very difficult to cross with the members of primary gene pool. And even if crosses progenies, I mean, if you cross and get the progenies, they are sterile and need special techniques for crossing. So gene transfer from GP3 to GP1 are extremely difficult. Right now, you have to try with secondary gene pool first, and then from there you can transfer that. All that you have to do. It's not easy. If you classify the germplasm like primary gene pool, secondary gene pool, and tertiary, these are the definitions and how they behave and what exactly it is. So agencies or institutes collecting and conserving the germplasm, or like IPGRI, International Plant Genetic Resource Institute, Rome and its network world over. AVRDC, now it is World Vegetable Center, now it is concerned to us in Taiwan. World Vegetables are renamed. The National Bureau of Plant Genetic Resources, NBPGR, what we have in our country. Like there are 4 sub-centers, remember, the Simla, it is representing the temperate zone. Jodhpur, Rajasthan, that's arid zone. Kanyakumari, Tamil Nadu, tropical zone. Amravati in Maharashtra, mixed climatic zone. So that represents. So that's why these centers are, located in different parts of the country to represent, and it is easier for maintaining the germplasm near to their place. What kind that particular species requires. Then you have got Institute of Plant Industry, Leningrad, what we said it way below. Russian, a lot of fragmentation, a lot of disturbance there. And then Royal Botanical Garden, Kew in England is also having a lot of variations. Then other institutes within India and world, they collect and maintain the germplasm, where you can seek the germplasm. Then FAO, Plant Production and Protection Division, with the Hortivar, they got it, and you can just get it there. Then the purpose and the function of the gene banks, why the gene banks are there, and what do you expect them to do it? First is collection and maintenance of germplasm. Second is evaluation of genetic stocks. Third is documentation of the germplasm. Fourth is multiplication and integration and renewal of seed stocks. Fifth is distribution of the promising cultivars. These are 5 important functionary of these gene banks. It lies. And so collection and maintenance, the purpose of germplasm collection is you can introduce it as a new crop. Maybe that area you don't have the crop at all, you can try, or it may be a new variety, or there's some existence you want to use it for crop improvement of that particular crop. So that is, and of course it's not only that, save the crop from the pest and disease. Sometimes some endemic situation comes, the entire crop, the species can wipe out. So by collecting the germplasm and maintaining in different place, you can going to save the crop from the pest and disease, because completely controlled conditions you can do it. And of course, the whatever you collect, this may be used for scientific study or some aesthetic purpose also. And then the types of collection, as per Herlan and House, it is accession collection, 2 sets, all types you are going to collect. Basic collection is a working collection. Some plant breeder wants to have the material to start with. Then spontaneous collection, it is not intended. Suddenly you get something very important. Wild and weedy allied and races should be maintained separately as source of resistance because of a handling problem. So whenever something very interesting, you can collect. That is spontaneous. And then the bulk collection, you are always having the breeding programs where a lot of material you are using and making the selection. So leftover material is there after making the selection. Leftover product of breeding selection, compositing close to the area of their adaptation, their bulk collection has to be. Leftover material, you have to maintain that. You are not going to take more care maintain that, but you are raising that collection near to their place of adaptation. That means where they are those species are originated, like that. So you can make your task easier. Then of course, named variety collection. All cultivars named released over the years. This includes the local and land races. Yesteryear, the local types were there, land races were there. The farmers were going on. Now, later, some new varieties came, then they disappear. That also has to be collected. And you release variety today. After 5 years, it may not be there, but that also has to go as a collection. So it's a continuous. Whatever released varieties, local types, land races, you have collection. Then the gene stock collection. So the stocks of known genes, that means one particular exon is there that is having a novel character, maybe menstrual gene it is having, or some anthocyanin pigment having gene is there. That is known as stock, right? Some exon known for a particular gene. Stocks of known genes, maybe translocation also, reversion, addition, or substitution. Nice, like the chromosomal aberration happened, but still you don't find that regularly. But that particular genotype it was observed, that you have to maintain special. So that it is called the gene stock. All right, maybe the reversion, addition, substitution, and even resistance or tolerance to known races of pathogen or pests, etc. So gene stock collection, that is known character having accession. Okay, so that is then the population collection. Carefully constituted population should be established and can be maintained. It is not immediately you are going to release variety from that. It is a continuous process. You are maintaining it. So byproducts can be there at any time. So some accessions you can push for your immediate breeding programs. So population collection. So those are the different 7 types of collections. Yeah, you have to carry out the exploration. These are the trips for purpose of collection of the various forms of croplands and their related species. Primary sources based on the sound of knowledge, biosystematic of genera and species to be collected, botany and taxonomy. That means before you go for exploration, the team, at least one member, should be having the idea about this. Biosystematics of genera, species to be collected, botany, taxonomy, everything. What are the related types, when they are going to flower, what kind of variability is there, idea should be there. Then ecology of the vegetation types of the place. What kind of ecology is prevailing where you are targeting this exploration. Then the climatology of the region, what kind of climate it has got, what kind of the temperature variation around the year, humidity variation, when it is going to have the rains, all that you have to. Then preliminary survey, before doing the serious exploration, some preliminary survey, just go around. So it will give you some idea. Then you can come back and plan your actual exploration. Then flowering and fruiting period is very important. Simply going and what you collect. So important characters are related to flowering and fruiting, when it flowers and fruits, and when to collect. You may be collecting the fruit or seed. So if you know the period, when in the year, what is the time that you're going to get that, so that time you have to, should know, then only you will be able to collect. Task is to collect as many diverse types as possible, without overcrowding. Some poor, simply collecting so many things, it's overcrowds, and some important things will miss if you have too many collections. So collect as many diverse types. And any next collection is there, it should be different from already what you have collected, like that. So without overcrowding, germplasm collection, and then often phenotype is not a good predictor of genotypes. This is also the explorer should have the idea. What you see, it may not be genetically controlled. That's of no use. So, but you depend, but you should have the idea of genetics prevailing in that species. Then it will be, better for that explorer to whether what you are seeing is real genetic variability or just masked by the environment. That's very important. Often phenotype is not a good predictor of genotypes. True. The forms of material to be, what are the materials you are going to collect? You can collect seed, you can collect a runner or cutting, tubers, palms, or fruits, or a pollen sometimes, right? You can, in the farm, you can collect from that place where the variability is there, or places where you're planning your exploration. So that is the first, germplasm collection. Then the second task of that, gene bank is the conservation of germplasm. That is storage, where to store. In situ, gene sanctuaries you can have. Ex situ, like a normal, the ex situ means, so normal temperature, lower temperature, prayer preserve, where you want to preserve, right? In the living gene sanctuaries means it is live maintenance, whereas ex situ means you are preserving seed or some material under normal temperature you want to preserve without losing its viability, or at low temperature, or sometimes you have to go for correction. As you reduce the temperature, the storability or multiplication in the frequency will be reduced. Yeah, the seed sanctuaries are important to maintain the germplasm alive. It has been proposed that within the centers of origin, areas of greater diversity should be demarcated. Diversity, we are mentioning a continent or a country, but some specific spots you have to identify. So that's what. Within the centers of origin, areas of greater diversity should be demarcated and protected from human disturbances. In such areas, the evolutionary potential of the local population and the environment would be preserved naturally. So that place is, you refer it as a gene sanctuaries. For example, citrus gene sanctuaries maintained in Meghalaya, northeastern region. You will find the gene sanctuaries of musards, banana, citrus, oriza, rice, and then mango. So gene sanctuary may be defined as the area of diversity protected from interference from man. Right? There it is showing the maximum diversity within the centers of origin. Okay. So that is conservation of germplasm. Then the evaluation of germplasm. Evaluate for quantitative character. It is going to be a very cumbersome process. Now I have to identify which are the characters you want to. That's why descriptor is available. What the curator, the person who is maintaining germplasm, can take observations there. Then whatever those the parameters recorded, the quantitative traits, rather it's a qualitative trait. So control by few genes, such characters is better to record. Quantitative, no use, because not having the identity, more of environmental variations there. So now those collected, whatever information you have recorded, that has to be documented. Completion of the plant descriptor. It starts from the beginning of the collection and completion of the evaluation. Sometimes you give the germplasm to some. Many of you students are getting germplasm from NBPCR. Now after evaluation, they ask you some data, how it has performed, few parameters. So it can be fed back from the place where it is evaluated to the gene bank, so that software, that data can be completely updated. Most of the seeds lose viability. Okay, that is okay. That is it starts from beginning and collection and completion of evaluation. Now the fifth responsibility, the gene bank, it's multiplication and introgression. It's very important. Multiplication, because the germplasm, if we keep after some time, it loses the viability. It's a dead seed or material. So you have to renew it. You have to grow it again. Most of the seeds lose viability quickly. Consequently, germplasm collection have to be grown every few years, depending on species. It's going to be a costly affair, requires much time, labor, land, and money, because so many collections are there. Maybe you are having so many crops to be preserved there. Resources always you should think. There is also the risk of erosion, labeling, and handling, something we are going to miss doing. But you have to do it. So that means with the very optimum frequency. The genotypic constitution of the entries may also change because of this multiplication, and it is true in cross-pollinated species and land races of self-pollinated species also. So you may lose what you have collected, and if you multiply for first time, what you have collected is going to be different. So these are all the negative points of multiplication, but still you have to. That's why don't do it every year. You do it with all the knowledge accumulated in the given species. How often we have to do it. So critical stage or a period needs to be decided. So just one more thing is introgression. So what do you mean by the introgression? Allow the wild species to cross with improved variety to favor the introgression. So what do you mean by the introgression? How you define? Introgression means the infiltration of genes from one species to another via partially sterile F1 hybrid by repeated backcross and selection of well-adapted backcross lines. So that means it's not easily. Introgression means from one species to another species. Genes transfer, infiltrate from one species to other species via, it's not easy or easily cannot be crossed because two different species means. So partially sterile, completely fertile, then no problem. Whereas sterility is there, completely sterile, no use. You cannot do it. Partially sterile, most of the time you get it. So you have to attempt so many and then few you get like that. The partially sterile F1 hybrids, and to regain, they have to go for repeated backcross and selection of well-adapted back lines. That is known as introgression. So infiltration of genes from one species to other species, that is possible when the hybrid is partially sterile and it is further repeatedly backcrossed and selections are made for which, the well-adapted backcross lines which are adopted. So that is entire process is known as infiltration. It's very important to germplasm value can be, enhanced by this. Naturally, it is happening, and you have to provide opportunity for that, growing wild species along with the cultivated species. That can happen, but gene sanctuaries, definitely it can happen. So the last responsibility of this gene bank is distribution of promising cultivars. So plant descriptors, if you look at, it is having the passport, extension data, and collection data, and then the characterization, preliminary evaluation, site data, plant data, vegetative, inflorescence, fruit, seed, further character and evaluations, like again, further you have to have the site data where you are evaluating plant data, vegetative, since fruit, seed, all those parameters, stress susceptibility, pest and disease susceptibility, cytological characters, anything. So now we should see that the plant descriptors available for a few of the crops with us. Passport data, access identifier and information recorded by collection. Then the characterization, when you said it, recording those characters which are highly heritable, can easily seen by eyes, and expressed in all the environments. Then the preliminary evaluation, recording of limited number of additional traits, thought desirable by consensus of users or plant breeders. Characterization and preliminary evaluation is done by the curator, whereas further characterization and evaluation should be carried out by the plant breeders, but they have to give the data back to that curator. So following internationally accepted norms for the scoring or coding of descriptors is done by and large. Measurements in SI system, the units applied are given in the square bracket followed by the description. And always description is going to use 1 to 9 scale. Any character if you take, maybe a disease or any quantitative character. And the description from 1 to 9 scale, right? Then the presence of the character is mentioned as plus. If character is not there, 0. So like non-tuberization, spininess, like that example. So if no tubers, 0. Tubers are there, plus. Then further, the characterization. So spines also. Action is not uniform. Mention mean and standard deviation where the character is continuous means mean and standard deviation where it is mean and chi value for the character discontinuation. If the descriptor is inapplicable, then you can just mention 0. So action is not flowering, you can't help it. You can't do anything on the flowering characters. Blanks are used for information yet not yet available. Standard color chart should be used, instant color chart or Royal Horticulture Society color chart, and date should be expressed in the first 2 digits days and next 2 digits months and last 4 digits year. The last part of this introductory first unit chapter, importance, various other related issues to vegetable breeding. That is a plant introduction, right? It's not really referred as a breeding method, but yes, it is one of the important activity If you don't have any organized breeding activities in your country, the plant introduction is very important. So plant introduction, it consists of taking a genotype or a group of genotypes of plants into new environments where they have not been grown before. Any kind of new environment. You can also see that new varieties of crop already, maybe it's not just, the entire crop you are going to introduce. I mean, the new crop, it is new variety also. The new varieties of a crop already grown in the area. That means the crop is being grown in this area, new area, but this variety is not. So introduction of new varieties can be done, or it can be a wild relatives of the crop species. And you can also target the totally new crop species for the area. This crop never grown in this area, but you want to introduce, that is also possible. So that kind of activity can restrict. Often materials are introduced from other countries. Normally introduction, you refer it for the one country to other country, or it is from one continent to other continent, or within country also it can be wide, because the movement of crop varieties from one environment to another environment within a country is also referred as introduction, totally a new environment. So you are referring within the country also it can be introduced, the genotype. So within a country we have examples like grape to Haryana. It was after, where earlier South India it was grown, or other states. And wheat to West Bengal, it was only the rice bowl at that time, but later it was introduced. Rice also introduced to Punjab, kind of introduction. Cabbage and cauliflower, they are temperate crops, but they were introduced. I mean, they were there in the country. Britishers introduced, but from the north to south. South is the warmer climatic condition having part in the country. So introduction of this cabbage and cauliflower to south is also referred as introduction. So introduction, you can classify, you can refer it as primary introduction or secondary introduction. Earlier, there was a plant introduction division at IRI, knowing the history. Said later, the plant introduction division, they started at IRI, and so many varieties were introduced from other countries. But later, this primary, the functioning was transferred by having the separate institute, National Board of Plant Genetic Resources at New Delhi. So what is primary introduction? When the introduced variety is well suited to the new environment, it is released for commercial cultivation without any alteration in the original genotype. That's referred as a primary introduction. This is less common in countries having well-organized crop improvement programs, because you may alter, you have got very organized breeding programs. So straight away, whatever the genotype you introduce, it cannot go as a variety. You can use that for crop improvement, and then later you release. So that's why it is said this is less common in countries having well-organized crop improvement programs. So, for example, you have got Sonara 64, straight away this variety introduced into India, dwarf wheat, and it was released for cultivation. Taichung native 1, rice, Bonneville tea. Now also you can find California winter, now also capsicum. Now it is reduced, but still you find these named varieties introduced from USA and other countries. They were straight away grown with the same name and without any alteration in the original genotype. So patty pan, still you can find the seeds in the market, summer squash. So that's primary introduction. Then the secondary introduction, the introduced variety may be subjected to the selection to isolate the superior variety. That means whatever introduced material is there, that is used in your breeding program. And then new varieties released. So alternatively, it may be hybridized with local varieties or transferred one or few characters to this local varieties, and then that local variety released as an improved variety. So this is referred as a secondary introduction. That means straight away it is not released. It is used in crop improvement program, and using this introduced variety, the new variety is developed and released for cultivation. This is much common in the then the primary introduction in most of the countries, right? So, example, Kalyan Sona, Sonalika, wheat varieties developed from material which were introduced from cement. These new varieties with our own name, they have, released. So, the material, whatever to develop this Kalyan Sona was introduced. So, for example, tomato, Pusa ruby, it is a selection from a segregating population of Sioux into Meruti. Meruti was a local type and Sioux was the introduced one. Arkavica selection from Tip Top, that variety introduced from USA. Straight away, Tip Top was not released for cultivation here, but using that, the selection was subjected for this variety, and Arcavicas was released for cultivation. Like you can have plenty, like capsicum Arca Basan pure line selection of Hungarian variety Sorry. Then Arca Gaurav pure line selection of golden calendar from USA, and Arca Mohini pure line selection from the Titan variety introduced from USA. So then radish you got Arca Nishant improvement from the Singapore collection IHS 72, or watermelon you have got Arca Jyoti F1 hybrid. It is a selection from Rajasthan local into Crimson Sweet. Crimson Sweet was introduced from USA. Okay, that's what the primary introduction and secondary introduction. What is the procedure for a plant introduction? You cannot straight away bring the material and release it to cultivation, or you can use it for crop improvement. But there is certain set procedures, especially if you are introducing from other countries. So procedure for plant introduction: first the procurement of germplasm, then quarantine. You have to separate it, keep it away, cataloging, evaluation, multiplication, and distribution. These steps you have to follow. Procurement of germplasm means ABRDC, Taiwan, germplasm of tomato, Chinese cabbage, sweet potato, several materials you have to introduce. So National Horticulture Research Institute, Nigeria, you have to identify which are the places from where you can get the material introduction. The National Horticulture Research Institute, Nigeria, and tomato, okra, and amaranthus. Central Horticulture Development, Senegal, tomato, French bean, and okra. Tropical Institute of Agriculture, Puerto Rico, tropical leafy vegetables we can introduce. First, locate where the material is there, and then we have to follow the procedure for introduction. Right? There is a procedure. You have to get the license straight away. You cannot. You'll be liable for punishment if you straight away get the material from other country. So yeah, you get all that, and through NVPGR, it is the channel from which you can introduce the variety. Quarantine is the next step after introduction. That straight away you cannot go use it for cultivation. What do you mean by the quarantine? To keep the material in isolation to prevent spreading of diseases, pests, weeds, etc. Any contamination you want to avoid. So first you have to keep it in isolation. Material is grown in isolation to record observations on diseases, insect pests, and weeds, and observe that how the introduced variety is carrying any pest, disease, or whether it is having any weed seeds and all that. So it will be subjected to quarantine rules. Rules prescribing them are there, like they call that quarantine rules. Whether this can be introduced or not, what specific procedure for this crop, it depends on the crop and country, right? Those quarantine rules are there. So all the materials being introduced must be covered by an authentic phytosanitary certificate from the source country. So from where you are getting the material, that country should issue the phytosanitary certificate. That means free, it's pure, no contamination. So they must be declared free from diseases, weeds, and insect pests. It's very important. That certificate it should carry. Then the plant materials accompanied by phytosanitary certificates are thoroughly examined. Right? It's okay. It should carry first. If there is no certificate, then reject, send it back. After that, if it is having the phytosanitary certificate, you have to examine. There are, quarantine people are there on that. They do that. Then materials contaminated by diseases or pests are destroyed or returned to the center if you find after examination. Fit material, that means you're not able to find any contamination. Yes, it is confirmed to quarantine laws, or fumigated. Still, you don't believe what you see by your eyes. So they have to be fumigated against diseases and pests, and then grown in isolation for a critical observations and destroyed or used for farm. So maybe some pathogens are hidden in the seed, and once germinate, you may get that pathogen or any insect, proliferation here. If it is found, it can be rejected. If it is not, then it can be further used. So there are, set of current and rules. I told you that those set of, the conditions you can just go through. It's being amended from time to time from the government of India. Restriction on export and introduction of plant materials. Not only just to get material, for sending the material to other country also there are set of laws. There is a restriction for export and import. Introduction to India is banned for cotton, berseem, sugarcane seeds through letter post. You cannot get it through letter post. And then banned crop and country. Cocoa from Africa, Ceylon and West Indies. Sugarcane from Australia, Fiji, New Guinea. Rubber from America and West Indies. Sunflower from Argentina and Peru. Because already we have, that's a dangerous. Any kind of quarantine you do, it's difficult. That's why straight away it is banned. You cannot import these crops from those specific countries. The ban on export of material from India, we cannot export like tea, jute, and black pepper because of the certain problems prevailing in that crop in that country. So that is the quarantine procedures. Once material passed through that quarantine, then you can catalog. Introduced material is given entry number. Exotic collection EC, indigenous collection IC, indigenous and wild. So these normally, NVPGR is giving you this prefix like EC, IC. EC means exotic collection, IC means indigenous collection, IW means indigenous wild collection. And then, okay, that's cataloging. And that means naming and giving whatever information is available that material. Then you need to go for evaluation to check the purity and check the purity of that introduction. Purity, it depends on the crop and extent of crossing. That limit is varying. So needs acclimatization. The whatever introduced variety, it may not be straight away fit. So the process that leads to adaptation of variety to a new environment is known as acclimatization. How to get it acclimatized? You have to spend time. That variety has to be, that introduced variety has to be grown here. It takes some time. And you need, and you need to do that. Grow in different locations you need to do. At one location, grow in 2 or more years and study stability performance. Then only you can inform. Then the last step is the multiplication and distribution. So once that is, past all these stages, if it is fit found, you can release directly as a new variety or give it for the crop improvement as a source of variability. So varieties, some examples, varieties introduced into India: tomato, Sue from USA, Marglo from USA, peas, early badger from USA, Sylvia from USA, Arkell from England, and various other plenty you can get to know. Those are all. French bean, contender from USA, watermelon, acai amato from Japan, sugar baby from USA, watermelon. Then onion, early grano from USA, capsicum, California wonder from USA, yellow wonder from USA, capsicum. So some examples are there. Straight away, those primary introduction examples are there. There are achievements of plant introduction, you can say. This can be, as it is said, what for it is used. A new crop species introduced into India, potato. See, tomato you can have, right? Potato, maize, groundnut, chili, guava, grape, pineapple, papaya are the introductions only. And recent introduction, 1960. After that, soybean, sugar beet, very fresh is oil palm. I believe that whatever the crop which is not originated in India, it has been introduced maybe a long back. Even tomato also, I told you, it was not at all a crop, before 18th century. It was introduced. Like that, any crop which is not originated, not having the diversity here, introduction. So plenty of crop varieties or crops are there. Crops, new crops have been introduced. Directly, you can, it's okay, that's a new crop. Then the directly released as a variety, Bonneville and early badger of pea and Sioux of tomato, straight away they have been released as varieties for cultivation. And varieties selected from the introduction, like tomato, Arka Vikas, capsicum, Arka Gaura, Arka Basant, Arka Mohini, then radish, Arka Nisan. And varieties developed, those are selection. Varieties selected from the introduction. Introduced material was subjected to selection. These varieties developed. Now varieties, whatever introduced, they will be used for hybridization program and then new variety. Like varieties developed through hybridization. Tomato, Pusa Ruby, Mirut into Sioux. Then Pusali Dwarf, Miruti into Red Cloud. Red Cloud introduction, Sioux introduction. Carrot, Pusa Kesar, the Nantis into local red, Nantis introduced. And turnip, you have got Pusa Kanchan, local red round, and Golden Ball. Golden Ball was the introduced. So like that, you can have plenty of examples for this, for introduction. It's not really a breeding method, but it is an activity, but going to give you the variety or new crop or the material for breeding. So with that, we are completing the discussion on the first unit, that is importance, evolutionary aspects, how it is different from the cereals, then centers of diversity or origin. More precisely, it is the center of diversity rather than calling them centers of origin, because we are not very sure that they have been originated there, because many crops showing variability in more than one place. Okay, centers of diversity and why they are important for crop breeding. It is the genetic variability. So genetic variability is the wealth of the crop breeder. What is his wealth? It is the germplasm. If he has germplasm, something he can do it. But then his ability to exploit it, and develop. So germplasm, collection, conservation, and exploration. Of course, the trips you are going to conduct, and then how to maintain that germplasm introduced and used it. And the very immediate process, after the, germplasm collection, something is the plant introduction. So we can use it as a new variety. So we have discussed in detail. So with that, we are closing on the first unit on the